prognostic markers. Actually, type 2 diabetes is different from type 1 diabetes in the sense that insulin is produced inside the body, but the insulin receptors are not able to, either they are deformed or due to some blockage, they are not able to detect the insulin molecules and carry out the process of glucose intake with the help of GLUT4 channels, and these channels and the cell is starved. In case of type 1 diabetes, insulin is not produced and the receptors are intact, and in this case also glucose is not taken inside the cell and the cell is again starved, and in normal case both insulin is produced and insulin receptors are active and glucose molecules are taken inside the cell and the cell gets its energy from that. Okay. So why it is important to study type 2 diabetes or type 1 diabetes, both diabetes? Because apart from increasing the glucose level inside the body, these diseases are also responsible for macro and microvascular complications, and which will lead to blockage of blood vessels as well as microvessels inside the body, and it will lead to diabetic nephropathy. Nephropathy means nephrons will start working due to blockage, due to high sugar level. These blockage will occur, and diabetic foot will occur, glaucoma, cataract, diabetic retinopathy, peripheral artery disease, heart attack, and stroke. And peripheral neuropathy like numbness inside the legs, etc. So it is very important to study this disease so that we can see that what changes are occurring inside the body, and the person will be more prone to heart attack or any other kind of risk factors associated with diabetes. Okay, so it is important to next. Why India should study it more and more? Because after China, India is the most important study point, or it is the hot spot of diabetes, and 74.2 million people are having diabetes in 2021. It is the last report which has been published by IDF, International Diabetes Federation, and in 2045 it is considered to increase up to 124.9 and even more. So mortality will be also there, and at very large level mortality will be there, and it is the data which is proposed. After seeing the lifestyle and all, it may be more, the figure may increase more, and this day by day is increasing more because of lifestyle as well as lot of pesticide related food. Pesticide is being used in crop production, etc. And junk food is also being eaten. So it is important for India to study. Now we come to the report. Actually, our lab, means the lab, molecular and human genetics lab of Lucknow University, we have seen the prevalence in North India is about 3 to 6%. Okay, and in South India the prevalence is shown more. You can see the red areas and the pink areas are having more prevalence in South India because they are studied more. They have studied more about type 2 diabetes, and in North India, few labs are only studying about type 2 diabetes, and most of the type 2 diabetic patients are hidden. Means they do not know that whether they are having diabetes or not. Okay, so that is why it is important to. Now I have selected to study this antioxidant genes. Why it is important to study antioxidant genes in type 2 diabetes? Because apart from most of the disease, cancer is also related to antioxidant gene levels. In type 2 diabetes, you can see that free fatty acid and hyperglycemia. If we take high rich fat-rich diet or protein-rich diet in most of the cases, which is required by our body. If we take more than that, then it is usual that we will have free fatty acid and hyperglycemia. In that case, what happens? Reactive oxygen species and nitrogen species are generated more and more, and oxidative stress occurs. And what happens after that? The NADH activity will be increased more from the required level, and after that O2 generation, free oxygen species will be generated more. And we can see that when oxygen species are generated more, the exocermin flux sorbitol pathway, PKC and H pathways have increased level of these enzymes related to that, and after that accelerated arteriogenesis, impaired vasodilation, lipid peroxidation, DNA damage, and protein nitration is caused by free oxygen species, and due to which diabetic vascular diseases will occur. That is micro and macro complications. This is the pathway which is related to reactive oxygen species, so it is important to study reactive oxygen species. So I have taken about 968 samples in which 558 samples were of type 2 diabetes and 410 samples were of controls, that is normal individuals. And the anthropometric measurements were taken of all these samples. Anthropometric measurements in needs, age, sex, BMI, WHR. Biochemical analysis was done and glucose level, lipid profile, and serum creatinine was seen in this. After that, we have done genotypic analysis. Enzyme assay was done of glutathione, superoxide dismutase, and catalase in few patients only. After that, genotype, allele carriage rate frequencies, gene interaction, and PDB analysis was performed. Following exclusion and inclusion criteria was taken for these patients in having North Indian ethnicity. All the patients were collected from KGMU, so they were having North Indian ethnicities. Ethnic group, they were of North Indian ethnicity. This is the list of, you will see that primers and the enzymes and where they are located. BMI was found to be important when significant association BMI is shown in non-hypothesis. All the biochemical parameters have shown significant association with type 2DM cases. When we talk of enzyme activities, significant association was shown in GST and SOD overall. After that, when we have seen GST, SOD and catalase, in case of females, all have shown significant and highly significant association. In case of GST, males have shown highly significant association with enzyme. So enzyme activity is related to, and when we see GST mean on all age groups, it has shown, GST has shown significant association, and catalase has also shown significant association between age group of 20 to 40. So we come to polymorphisms. These polymorphisms have been carried out. First is GST M1. In this, null and present. It is null and present, and these are the markers. Okay. CD36 control and beta globin control has been taken. These are the markers in which null and present. It is also the marker, and in null cases the gene is completely absent. So it has been done. Note in GST T1 also it is null and present. So what is the association? In case of GST M1, null gene has shown a high odds ratio in when cases and controls are compared. In case of T1 also, null gene has shown high odds ratio. Again, in case of P1, this is the digestion and genotype. So we come to the results. AG genotype has shown a high odds ratio and p value is highly significant. Okay. This is catalase gene graph. Again, AT has shown high odds ratio and significant association in case of catalase minus upstream A to T polymorphism. All the genotypes have shown significant association with the biochemical parameters, which include creatinine level, ADMR, waist hip ratio, fasting, postprandial glucose level, triglyceride level, total cholesterol, HDL, LDL. Okay. So means biochemical levels are already established. Again, we talk about SOD plus downstream 47 C2T polymorphism. Okay. In this case, C negative, when C is absent, then it has shown significant association and high odds ratio. So what GTX has again shown, CT genotype has shown highly significant association and high odds ratio. CT genotype is connected with the disease prognosis, and again this is the biochemical level. So haplotype analysis have been used here to show a statistical correlation between gene in two different genes, as it is a software which is generally used for haplotype analysis. But in this case, gene interaction analysis has been also used, and we can see GST-P1 and GST-P1. GST-P1 has shown high significance. Okay. So now we come to the group of genes which we have taken. So in most of the cases, like GST-M1 and GST-T1 has shown high odds ratio. GST-M1, T1, P1 has shown NNT genotype has gene combination, allele combination has shown 13.473 means odds ratio, which is very high. The highest has been shown with this case. You can see this one. IPP means present, GST-M1 present, GST-T1 present, and isoleucine, GST-P1 isoleucine amino acid, T, C, and T nucleotide have shown a odds ratio of 5083.345, which is quite high. Okay. So only one family I have taken to discuss. You can see that in case of first generation, female individual has shown NNVTCC genotype. Means an first individual has shown the significant means genotypes which have shown high odds ratio in this case. So we can use these as we can conclude that TTC, apart from the individual genotypes having significant association, we can use TTC which is 13-point high disease risk, and we can see MPIACT allele combination which has got 2.5 to 5 times disease risk, PPI-TCT combination which has got 5083 times high risk of developing type 2 diabetes. In prognosis as well as we can see those individuals who are young and not having disease, and we can check their genotype and say that they have got high risk of having type 2 diabetes. Thank you. This was my presentation, and all the work has been carried out under the supervision of Professor Manisha Banerjee, and I am here to present. All work is dedicated to her only. So, any questions?
I think you have done the genotyping. You must have understood better because you have already done that. If you cannot, did not follow anything, you can ask sir also will be there.